The adult stem cells have potency to generate every cell types in each tissue. So for example, the adult stem cells in the brain can generate brain cells, and adult stem cells in the intestine can generate intestinal cells. If we think of our body, the organs seem to completely formed without any change, so one may think we don't need stem cell population in our body parts. However, if we look into the tissues, for example, intestinal epithelium, most epithelial cells actually renew every week, so stem cells have to constantly generate the new cells. Adult stem cells are also important for the regeneration. They are the core parts to generate new cells after cell damage. So it is not an overstatement that we cannot live our daily life without our adult stem cells.
So one important bit what we can learn is how actually our organs develop, and more specifically, not just to observe how the organs develop, what we know by now, but to really understand what are the underlying molecular mechanisms. So which factors are actually involved in the whole process. The guiding principle, or the underlying basically logic of this, can be sort of summarized, or like a famous physicist, the Nobel Prize winner Richard Feynman, actually nicely summarized it when he said that what I cannot create, I do not understand. That means if we really truly want to understand how a process works, in this case organ development, we should be also able to recreate it in vitro. So we recreate it in the dish in our labs. And that gives us a huge advantage if we can recreate that process, because then we can also analyze it in more detail using methods that we cannot use, for example, on embryos or on patients, etc. That essentially means that we try to recreate human development or human organ development in a dish, but also disease state in a dish. And that also means then we can then go on to develop new therapies that are much more targeted for a particular organ, in our case, for example, the heart, and that we also understand much better what, for example, the side effects can be, and also to mitigate for these side effects. So that, I think, would have a huge impact on our knowledge and also how we do medicine in the future.
Okay, it's a very interesting question, and as a stem cell biologist, I think we think about cancer differently from the cancer biologist, because in case we have a mutation in differentiated cells, we all know that these cells are going to be lost from the tissue, so the mutation will be also lost. Whereas if we have a mutation in the stem cell, they will be spread to all the differentiated cell types that are coming out from the stem cell. And whenever we have an injury in the tissue, stem cell will work on to repair the tissue, and then the mutation that was in the stem cell will spread through the whole tissue. So this is a big problem, and I think understanding how a stem cell containing mutation is working before the tissue undergoes transformation is really important. And for that reason, our lab is utilizing a genetic method with which we can color a stem cell, at the same time giving a mutation. And by doing so, we want to track how the colored stem cell can transform the whole tissue in a given time.